In this video, we're going to cover the mechanism of transcription, which is a step in which information from DNA is copied as into RNA. So I'm showing here the steps in the transcription in bacteria, and the reason I'm showing this is because it's a pretty simplified way of showing the different steps, and really the important elements are present both in bacteria and eukaryotes. So we have, if you can imagine, you have the genome of any organism, and I'm going to draw it as a straight line. And within these double-stranded molecules of DNA, which makes the chromosome, are sitting genes, each of which code for a particular protein. And then in between these genes are sequences that are not genes. So in order to make an RNA from this DNA, the enzyme that is involved in making RNA, which is called RNA polymerase, it needs to know which pieces of DNA need to be converted and copied into RNA. And how does the RNA polymerase recognize that? It comes down to specific sequences that are present on the chromosome itself. So let's just move down and zoom in on here on one of the genes and see what happens. So if we go back, I'm going to keep my coloring consistent. So let's say if we were going to zoom in this area, okay, so then we have the gene sitting here. Let's say this is a gene for codes for some protein, let's say actin, a component of your cytoskeleton, whatever it is. Okay, so this gene needs to be copied into an RNA. But how does the RNA polymerase know where to find it? And it turns out it can find it by a particular sequence that sits before the gene starts. Usually it has a signature sequence, but I'm not going to go into it because it's at this point it's really not important. And this sequence basically its job is to recruit RNA polymerase. So as an analogy, imagine that you in your complex that you live, there's multiple houses, you have a birthday party, and how would people know, how would your guests be able to find your house? In addition to give them an address, but sometimes it's house numbers are not immediately obvious, the address might not be straightforward, so what do? You put up balloons outside of your house to say, hey, the party is here. So you can think of this sequence as providing something similar to that. And we call this sequence a promoter that I've written here. So this promoter its job is to recruit RNA polymerase. But it doesn't recruit RNA polymerase directly. It actually recruits proteins called transcription factors. Now in bacteria, the transcription factor is called the sigma factor. In eukaryotic cells, there's different names which I'm not going into, but the point is this promoter recruits a protein, recruits a transcription factor, okay, which then this transcription factor recruits the RNA polymerase. So that's shown here as a triangle. So first the transcription factor is recruited, then that transcription factor brings in the RNA polymerase. So the role of the promoter is to recruit the enzyme that is required to make RNA. So once the RNA polymerase is recruited to the DNA, we say that the first step of transcription has completed, and that's called the initiation step. Transcription has initiated. Then the RNA polymerase opens up the DNA and starts copying the information on the DNA. Now, unlike replication, where DNA polymerase would need a separate molecule like helicase in order to open up the DNA, RNA polymerase doesn't need that. It can do that on its own. So then RNA polymerase comes in and then opens up the DNA and starts making a copy of the sequence of DNA. And the principle is basically the same. It's based on ability of nucleotides to complement each other with respect to the bases that we have. So hydrogen bonding between bases is the basis of copying the DNA. So as we know, A, hydrogen bonds with T, and then there's 2 hydrogen bonds, and then G, hydrogen bonds with C. Except when you make RNA, you don't place a T in the RNA, you place a U. Okay? So then the information is copied. And also notice another thing that is different as compared to replication is that only one strand is being copied. The other strand is not being copied. So in transcription, the strand that is being copied, we call it the template strand, because it acts as the template for making the RNA. So the template that is being copied, we call it the template strand. And the other one is the non-template strand. Okay, so the RNA polymerase goes along the DNA, makes the copy, but then there needs to be some signal telling the RNA polymerase when to stop making more RNA, because there is an end to the gene. And that is determined by sequences that a DNA sequence that is at the end of the gene, which is called the terminator sequence. So once the RNA polymerase reaches the terminator sequence, it knows when to stop. So before the RNA polymerase reaches the termination sequence is called the intermediate step, is called the elongation phase of transcription. But once it reaches the terminator sequence, then it lets go of the DNA and of the transcription factor and of the RNA, and then the transcription stops and it's completed. So here's an exercise for you. Here's a DNA sequence. Label the DNA strand that you use as template to make the RNA.